morning, dear students. Today we are going to continue the remaining of translation. In the last class, what we have learned, we learned about codons, what is its nature, that is triplet nature, and what is the way of interaction between different tRNAs and the codons of the mRNA, what are the probabilities, all those things we have seen in the last class. Today we are going to see how that amino acid is added to the tRNA. Okay, that process is called amino acid activation, and which is the enzyme necessary for attaching the specific amino acid to its corresponding tRNA. The enzyme is aminoacyl tRNA synthetase. In a cell, there are 20 different aminoacyl tRNA synthetases. Why? Because only 20 amino acids are there to get added to the various tRNAs. For creating almost all available proteins, only available number of amino acid is 20. So corresponding to that, 20 different enzymes also present. Then how that addition of the respective amino acid happen to its corresponding tRNA. First, I am drawing the structure of tRNA. This in this way. tRNA. Oh, look. This is the acceptor arm. Acceptor arm 3 dash. And here's the anti-codon sequence. We can take the case of enzyme alanine. Suppose we need to add alanine to its corresponding tRNA. For adding alanine, the enzyme will be alanyl tRNA synthetase. This will be its enzyme. Then how this alanyl tRNA synthetase recognizes its correct concerned specific tRNA? That only because of the presence of a specific base pair that is present in the acceptor arm of the concerned tRNA. By identifying that base pair, this alanyl tRNA synthetase adds its correct specific alanine amino acid to this tRNA. Clear? Okay. If we insert the base pair to some other tRNA that is specific to some other amino acid, for example, phenylalanine, then what will happen? This base pair will be taken from here and inserted to this tRNA. Then the alanyl tRNA synthetase add this alanine to the acceptor arm of this tRNA. So the primary determinant is what? A specific particular base pair that present in the acceptor arm of tRNA determines, that helps the aminoacyl tRNA synthetase to add its corresponding specific amino acid. Okay. Next is the reactions carried out by aminoacyl tRNA synthetase. This is the enzyme we know, and this enzyme is adding the specific amino acid to its corresponding tRNA. How that addition is taking place, that I am going to explain. This is the enzyme, aminoacyl tRNA synthetase. It picks what? Amino acid. After this amino acid bind to this enzyme, it is activated by one molecule of ATP. We know that ATP is nothing other than adenosine triphosphate. Okay. As the enzyme picks this amino acid, from this ATP, two phosphates are released. Two phosphates released. That means it is giving energy to the amino acid. It is energized or activated. And then that amino acid is in adenylated form. Then that amino acid we can call it as aminoacyl adenylate, that is bounded to the enzyme. Okay. Then what happens next? Now this amino acid is activated or energized. Then came the tRNA. How this enzyme recognize its complementary or correct tRNA, that I already explained you, by recognizing a particular base pair in its acceptor. Okay. And then this tRNA command binds with this enzyme. After that binding, the one AMP that's still present inside the enzyme is released. Okay. The tRNA binds with the enzyme, and that amino acid, activated amino acid, is attaching to the 3' acceptor arm of this tRNA. Then what next? Then that enzyme is released, and the tRNA with the amino acid also released. Then came another factor, elongation factor. That elongation factor command binds with this tRNA that is holding that amino acid, and fetch this tRNA to the ribosome that is carrying out translation process. This is the ribosome. This is the mRNA. This is the growing polypeptide chain. To this, this tRNA need to add this amino acid that it is bringing to here. So that tRNA binds to some specific site in this ribosome. Regarding that, I will explain to you later. Bind to that specific site. After it binds with that specific site, then that amino acid leaves from the tRNA and is added to the growing polypeptide chain, and after that, the tRNA is released from this ribosome to start or again to begin another round of the same process. Okay. Some other interesting facts also there in this translation process means if somehow if a wrong tRNA is connected with a particular amino acid, then what will happen? Then that enzyme means aminoacyl tRNA synthetase. That aminoacyl tRNA synthetase can recognize that wrong incorporation and can sever the bond that connect that amino acid with the tRNA, and can make and can correct that misincorporation. So that is one interesting feature that showing the important significance of that enzyme. Next, I am going to explain about two important factors that is very important in translating the nucleotide sequence in mRNA. First one is the enzyme. Enzyme, what is the name? Aminoacyl tRNA synthetase. And the second one is the anticodon that present in tRNA. Okay, what is the significance of this enzyme? That I already explained to you. That enzyme itself incorporate a particular amino acid to its concerned correct tRNA, mainly based on the presence of a particular base pair in the acceptor arm of this tRNA. That I already explained to you. That's very clear to you. Then we need to understand the significance of anticodon of tRNA in translating nucleotide sequence of mRNA. For that, a scientist, his name is Fritz Lippmann and his associates. They performed one experiment. He prepared a tRNA, tRNA that is carrying cysteine. Okay. After this tRNA got incorporated by this cysteine, the scientists altered this cysteine to another amino acid, that is alanine. Okay, but its anticodon remain unaltered. It is the same itself. So what happened? This anticodon recognized the same codon in the mRNA. mRNA. This anticodon recognize the same codon in the mRNA without bothering about the change happened to the amino acid. As a result of that, the polypeptide chain that formed, it possesses alanine amino acid in the place of cysteine. Clear? That means that amino acid has no role. The important thing is the complementarity between anticodon and the codon that present in the mRNA. That determines the amino acid sequence in the polypeptide chain. Okay. With this, we finish today's section here. In the next class, we will learn about translation, what are its different steps. Okay, thank you.